Can you guys hear me?
Yes.
Yep.
Okay, good. All right. Yeah, my audio didn't connect for some reason. How you guys doing?
Good.
Okay.
Good.
Good.
Did you guys get what was it? What was that, Kelsey?
I just said good.
Oh, good. Did you guys get the email with the old exam attached? Okay. Very different from tests that you've seen from me in the past, right? Okay. All right. So I just wanted you to have that so you kind of know what kind of things are coming. I started working on the test for Friday yesterday and didn't get very far because some other things came up during the day, but I'm going to continue working on that today. So if you haven't started studying for it, you should certainly start. The other thing that I realized that earlier in an earlier lecture, I had told you that the test was only going to go up through amphibians, the end of amphibians. And if we started on reptiles, that we wouldn't put reptile things on there. Do you guys recall me saying that? And then on Monday, I told you that the test would be over whatever we finished today. I'm going back to my original statement, which is actually how, when I went back and looked at that old test, I realized that the old exam one was structured the way I told you originally, which was to cover everything up through amphibians. So this stuff that we covered on Monday on the reptiles and we'll cover today on reptiles, that'll be on the next test. This test will stop at the end of the amphibian material. Is that clear? Okay, good. All right, before we go to questions from last time, I just want to tell you a couple of things that I found interesting in the time since I last saw you guys. I think one of the hardest things to do as a biologist nowadays is to stay current with the literature, simply because there's just so much literature coming out all the time, every day. And on that note, I was made aware, strangely enough, through social media, of a new paper that just came out that looks at Lyme disease and the fact that it's related to lizard abundance. So there have been some papers published on the relationship between ecology and the incidence of things like Lyme disease in human beings. So there was a paper that I use in some of my classes as an exercise to teach about food webs, where it shows that Lyme disease is influenced actually by masting in oak trees. What happens is when oak trees have a masting event, the oak trees produce a ton of acorns. Those acorns feed populations of mice and deer. The mice and deer then, because the ticks feed on the mice and deer, the tick population goes way up. And then the next year, the following summer, you get higher levels of Lyme disease being reported in humans because of this increase in tick abundance that is due to the previous year's increase in mammal abundance, which is due to the previous fall's acorn production by oak trees. So I've taught that kind of thing in the past. Can you guys see this paper from PLOS Biology? Yeah. So what has also been coming out lately is that in the southeastern United States, the incidence of Lyme disease is much lower than the incidence of Lyme disease in the northeastern United States. And it turns out that this is because skink abundance is much higher in the southeastern United States than it is in the northeastern United States. Skinks, basically, they have a distributional limit northward. And so in northern Maryland, you start exhibiting a decline in the diversity of skinks and the density of skinks in forests. And what these people have found is that Lyme disease is common in the northern US, but rare in the south, is mainly due to the fact that skinks are more abundant in the southern US than they are in the northern US. Skinks are a type of lizard. We'll start studying skinks after the test next week in lab, among other things. So it's a type of reptile, a type of lizard. And so I saw this yesterday, and I was talking to my partner at home last night. I'm like, oh, I saw this cool paper today about Lyme disease and skinks. Evidently, Lyme disease is less common in places where there are lots of skinks. And of course, what was her first response? What would be your first response when you hear this, having not read the abstract? Why you think Lyme disease might be lower in places where skinks are common? Anybody?
Sorry, did I not unmute?
Oh, you didn't unmute. Oh, I'm sorry. Yeah.
Oh, sorry. I said because they prey on them.
Okay, so that would be a logical thing. So actually, Elizabeth, the first thing she said, oh, do skinks eat ticks? I'm like, no, they don't. So now put on your critical thinking caps and say, okay, if Lyme disease is less common in the South because there are a lot of skinks, but it's not because the skinks eat the ticks, what do you think might be happening that reduces the incidence of Lyme disease in humans as a result of skinks being present?
Are the skinks eating the mice that are like the carriers?
They're not eating the mice that are carriers. Skinks are generally, at least in the United States, they're on the small side. There are skinks that live in Australia that are quite large and could eat mice, but I don't think any of our skinks, with the exception of possibly one species that lives here in the Midwest, they're really not big enough to do that. But that would be a good hypothesis also. Turns out not to be the case. What other things might you think about? Think creatively a little bit. In northern North America, so up in like New York and places like that, oak trees produce a lot of acorns. Acorns feed the mice. Mice populations go up. Because mice populations go up, the tick population goes up, and then those ticks ultimately get onto a human and then transmit Lyme disease to them. So what might be happening in a forest that is dominated by skinks? Because skinks can be really abundant locally. Think about parasite life cycles going through different hosts. There's a clue to this in the title of this article.
Do the ticks prefer to feed on these, whatever they are? What do we call them? Skinks?
Yeah, so what happens is in the southeastern US, where skinks are really common, it turns out that ticks parasitize skinks more than they parasitize mammals. And mammals are the type of intermediate host that Lyme disease needs to be in order to complete its life cycle. And as a result, when the ticks feed on the skinks, the Lyme disease vector, the organism that causes Lyme disease, finds itself inside a dead-end host. And so the host choice by ticks in the southeastern United States is more often skinks than mammals. And as a result, it kind of stops the transmission of Lyme disease. So they're not eating the ticks, they're just providing a different type of host for the ticks, and that disrupts the cycle that gets Lyme disease into human beings. And so, I don't know, I found that an interesting sort of indirect role of lizards. And strangely enough, as a result of reading this, I went back and found an earlier paper from 2011, where in the Western United States, they found a similar thing happening, not with skinks, but with the Western fence lizard, Scoloporus occidentalis. And so, and it operates the same way. The lizards aren't consuming ticks, but the lizards are in great enough abundance that they serve as an alternative host for the ticks. And that alternative host happens to be a host that Lyme disease doesn't. It puts them in this dead-end reservoir of the disease. And as a result, you get less transmission of the disease to human beings. So one of my goals in every class that I teach, because I tend to teach classes that a lot of times students don't feel that they need to pay any attention to, because what on earth does any of this have to do with humans? And I'm planning on going to medical school, so I don't care about anything other than humans. But we constantly, as we learn more and more about the natural ecosystems around us, the more we find that these ecosystems are in some cases tightly tied to issues of human health. So if you need another reason to think, oh, I need to pay attention and be engaged in herpetology, sometimes herpetology directly impacts things like human health. This guy, Richard Ostfeld, is one of the guys who did the work on Lyme disease in the northeastern forest, where he linked the production of acorns by oak trees to cycles of Lyme disease. He's actually used the models that they've created studying ecosystem level sort of processes, and basically has created a model by which epidemiologists in the northeastern United States basically make contingency plans to preemptively alter people's behavior when they can predict when there's going to be an increase in Lyme disease because of things that are happening ecologically. So if you live in New York, for example, when there's a masting event in New York where the oak trees are producing a lot of acorns, what the health departments will do in New York is, as the winter passes after that masting event and they're going into spring, they start to send out health advisories, basically telling people to either go hiking less often, or in these health advisories, they also give you lots of practical advice on how to avoid ticks if you're going to go out and go hiking in the Catskills or something like that. Once again, to reduce the odds of you getting Lyme disease, even if you're going to be out there, by basically encouraging you to take preemptive action against ticks during those years when Lyme disease is most likely to be more prevalent in the environment. So anyway, human health tie-ins between skinks and humans, and fence lizards and humans, and oak trees and humans. So, all right. Any questions over the kind of things we were talking about last time? Okay, so we were talking about skin and shedding, and how reptiles produce skin that exists in 2 layers over time, starting out as a single layer, this inner generation layer that will ultimately become the outer generation layer as these layers differentiate. And as the outer layer is protecting a developing inner generation, that inner generation then finally has matured to the point that it's created scales and sclerification, or the keratinization and things like that, at which point the outer generation is shed and then the inner generation becomes the outer generation. And so shedding occurs in the squamates. It comes off in these big patches. In crocodilians and turtles, it flakes off in smaller patches over time, but all reptiles shed. We talked about how these scales present a barrier to sensory perception, and then we were beginning to talk about other forms of sensory perception that are not necessarily inhibited by the presence of scales. We had talked about Jacobson's organ, which is the scent organ in snakes, and one group of lizards. And we talked about these lenticular organs, which I had already introduced back when we were talking about gecko toes, these sensory organs that are these thin layers of the skin that allow for the sense of touch through the skin. So reptiles can hear. They have a tympanum, so you're going to see ear openings on all of the reptiles with the exception of snakes. And so when you see, somebody mentioned back during the introduction, I forget who it was, they found it fascinating, these legless lizards that look like snakes but aren't snakes. They're just lizards that have evolved out of their legs. Well, there are a couple of ways of telling legless lizards apart from snakes. One way is that snakes don't have eyelids. They have a scale, a transparent scale that goes over their eye. So they're looking through this transparent scale. Lizards have eyelids that they open and close. And the other way that you can tell legless lizards from snakes is they have an ear opening, an external ear opening. And so they have a tympanum just like amphibians do. And these transmit sound to a cochlea much in the same way that happens in humans. Snakes lack a tympanum, but they have all of the internal structures of the ear. And what that tympanum does is, because the tympanum which is this thin membrane that is in contact with air, snakes can't pick up high frequency sound because they lack that tympanum, but they can pick up low frequency vibrations. And this is important because as you're walking through a forest or walking through a desert or something, a snake may be able to not hear you coming, but a snake can feel you coming because it senses these ground-borne vibrations. This was made evident to me back when I was in grad school, because one of the guys in my department was a cardiac physiologist. He studied heart physiology, and he used snakes as one of the groups of organisms that he looked at. And so what he would have in his lab is he would have a table, and on this table would be a cage, and in the cage would be a snake. And to the lab there was a door, and he had a window in this room so that he could observe the snake from the other room, but he would occasionally have to go in and do something with the snake. And one of the things, so mounted underneath this table was actually a sensory setup that he had invented, where the table was thin, and I forget the details of how it did it, but basically he had it set up such that it could read the heartbeat of the snake through the bottom of the table that the snake was sitting on. And one of the things that he noticed when he was looking at his electrocardiograms that he was recording is that even if he didn't interact with the snake at all, just the process of opening and closing the door made the heart rate of the snake go up, even if the snake was in a hide such that the snake couldn't see the door being opened. And he concluded that what was happening is opening and closing the door was sending low frequency vibrations through the floor that went through the leg of the table into the surface of the table. The snake was picking up those vibrations, knew that something was happening outside, even if it couldn't see it, and immediately started reacting physiologically in a way that it was ready to, for example, strike a potential predator or something like that. So he found that as a result of doing this, that anytime he wanted to do something with a snake, he had to do all of his preparations and everything, and then give the snake some time to kind of come down from that disturbance, even if he hadn't done anything in the room other than enter the room and exit the room, because the snake was capable of sensing these low frequency vibrations. And of course, that lengthened the amount of time that he had spent doing research with the snakes, because he had to give every snake time to kind of readjust after that disturbance. All right, vision. Most reptiles have decent vision, well-developed eyes in many cases. One of the distinctive features that is different about their eyes as opposed to ours is that they have these bony elements that go around the perimeter of the eye, roughly approximate to where the iris would be, and basically going up against what is the lens. So these things are called scleral ossicles, and they're found in all of the squamates. They have rods and cones just like you do, but they're somewhat different in terms of their structure. There are lipid drops in these cells within the eye that are responsible for responding to light, sending signals to the brain to indicate that light is there. And one of the things that these lipids do is they block shorter wavelengths of light. And so their vision is going to be different than your vision. They're going to perceive color differently because they have the presence of these lipid droplets in their rods and cones, or in the cone cells rather. So these are just some comparisons between the eye structure of lizards and snakes. Once again, this is a table from your book. I'm not going to require you to know this table. This is just for your reference. But know that things like these oil droplets do affect color perception, and as a result of that, the way in which snakes and lizards see the world is going to be very different from the way you see the world in terms of color perception. One of the things that you'll notice about a great many reptiles is that they are not particularly colorful, whereas many of the amphibian species that we looked at sometimes have very vibrant colors, and that tends to be less the case in reptiles. So once again, just basic eye structure. You typically have a cornea and a lens, and then a vitreal chamber where the vitreous humor is. And then you have the retina along the back of the eye, which is where the rods and cones are held. This fovea right here, I don't know if you guys have had much in the way of anatomy. This is a little blind spot that is in the eye. You have one also. It's a blind spot because this is where the optic nerve, which services all of the retinal layers, all of those optic nerves from the retina have to exit the eye, and they exit the eye in this one spot. And so that's where the optic nerve is entering, because that is a patch of the retina where it's basically made up of the dendrites of the nerve cells that are sensory nerve cells that are servicing the retina. There is a reduction in rods and cones in that area, and so you have this little area that is a blind spot. Once again, these scleral ossicles go around the eye at about the level where the lens is. Lizards have these scleral ossicles, and these scleral ossicles are not found in snakes. All right, in some groups of reptiles, in particular certain groups of snakes, they have pit organs. And these pit organs are involved in the detection of infrared light heat, basically. And so they are able to sense really fine grain differences in heat, and they use this for essentially detecting and tracking prey. These pit organs, which are in some groups found in their own special organ on the front of the face, just down from the nostrils, or they're sometimes found in either the scales of the upper lip or the lower lip. But regardless of where they're located on the face, they're all served by the trigeminal nerve, and they go and implant basically into the optic tectum of the brain. And the optic tectum of the brain is an area that essentially forms a spatial map. And so they can use information coming from these pit organs to basically determine where in three-dimensional space the heat source is located at. And so there are some hypotheses about how they developed this particular ability. And so there are actually people actively studying this particular feature of snake biology to try and figure out what genes are responsible for allowing this infrared detection ability. And so there are some thoughts about which types of genes are responsible for this. Obviously, this is one of the unique characteristics of snakes in the animal world. So here is that looks to me like a reticulated python. So in pythons and boids, you see that they generally have these thermal pits located in the either upper or lower labial scales. So this row of scales that goes along the mouth here are called labials. And in some groups, it's just the upper labials, and some groups, it's just the lower labials. But these open pits open onto a set of sensory epithelia that are once again tied to the trigeminal nerve. And so this is just branching trigeminal nerve that essentially carries the information to this section of the brain, which is where strangely enough, optical information from the eyes is being processed. And so these are called thermal pits. And in the pythons and boids, they're found in the labial scales. In pit vipers, there is a special pit on the front of the face. This is the nostril, this is the eye. There's a pit that lies between the nostril and the eye. And you'll also notice that it's kind of conical shaped and faces forward. This is just a blow up of what that looks like. And once again, this is the branches of the trigeminal nerve. And once again, when you do nerve tracing studies, you find that they go back to this section of the brain. So a little bit of critical thinking here. Can you guys see the board okay? Or do you have problems seeing the board?
Yeah, it's still blurry for me like it was before, but you know, it's fine.
It's not fine. If you can't see the board, then there's no point in using the board.
This is what I do. I don't know if you guys know how. If you click on Paul's screen on the Zoom, you can actually switch to the whiteboard versus the presentation mode, and then you can switch back and forth instead of you having to do it yourself.
Yeah, I did that. It's just sometimes it's still blurry because like my Wi-Fi doesn't connect to my computer well, but sometimes it's readable and sometimes it's not. It literally just depends on the day.
That's okay. I've learned some things in response to, you made that comment earlier, Rose. That's Rose, right? So I have since then gained new skills. Just give me a minute. It sometimes takes a minute for it to take effect. And sometimes I have to restart my Wi-Fi in order to get it to work. But that's easy enough to do. Can you guys see a blank screen now? Okay, so here is a snake head, crudely drawn. There's an eye, there's a nostril, there is a forward-facing thermal pit, a lower jaw. Head is attached to the rest of the snake. Now imagine that you're a rattlesnake or a copperhead or a cottonmouth or something like this, any of the pit vipers that we have in Missouri, and a mouse comes by. I'm going to draw my standard mouse. Little mouse ears, a little eye. And this mouse is emitting heat, and that heat is being picked up by the thermal pit of the snake. Now, when the mouse passes by the snake is going to strike. But when the snake strikes, the snake opens its mouth such that the eye is now facing upward, the nostril is facing upward, and this thermal pit is also facing upward. Yet the snake, with unerring accuracy, is capable of striking the mouse with its extended fang. That's a terrible looking fang. Let's redraw that fang. Strikes the mouse with its fang, in spite of the fact that it's pitch black outside because these things are hunting at night, and the thermal pits, which it uses to locate the mouse, are pointing now at the sky. They're not pointing at the snake at all. This is a question that guy that I know at Colorado University at Boulder had, which is basically if the thermal pit is an adaptation to acquire food, to detect and acquire warm-blooded prey by these pit vipers, how do they continue to localize prey? Because what you would oftentimes see is that when you have a snake laying on the sand or in the grass or whatever, and a mouse comes by, when the snake starts to strike, the mouse runs away, and you will actually see the snake turn in mid-strike to track the mouse. Once again, even though the snake's mouth is open and the thermal pits are not facing the prey any longer. So he had the question of how do snakes, pit vipers, track these warm-blooded prey when the thermal pits are no longer directed towards the prey. Any hypotheses? What are your thoughts?
Does that have to do something with the vibrations again?
It doesn't have anything to do with the vibrations, but that would be a decent hypothesis. And substrate-borne vibrations are probably going to be spatially non-specific enough that it would be difficult to detect the exact location, especially if a mouse is running for its life because it's about to get struck by a rattlesnake. I'm going to go back to the PowerPoint, because I think seeing the PowerPoint image will perhaps help you. Is the PowerPoint now on the screen? So you can see this nerve here that serves the thermal pit. What else do you see where there are nerves?
They extend towards like the nose and like the snout region.
So are you talking about these up here? Or are you talking about these down here?
Well, both of them, kind of.
Okay, so these up here are probably not as important, but these down here, it looks like, because this is like a transparent snakehead, it looks like they might be going to these labial scales, similar to these things that are going to these labial scales and in these thermal pits that are in the labial scales. But in reality, these nerves are going to the oral mucosa, the lining of the mouth. If these are going to the lining of the mouth, what might they be doing? Especially when you consider that this nerve is actually just another branch of the trigeminal nerve that goes to the thermal pit. So these are all three branches of the trigeminal nerve as it leaves the brain. What might these nerves be doing if they're going to the nasal mucosa of, for example, the roof of the mouth?
Like sending signals about the prey.
Yeah, so in the roof of the mouth, you have all sorts of sensory organs. You have, well, not in the roof of the mouth, but on the tongue and things like this, you have taste buds. But also in snakes, you have these nerves that are going to the roof of the mouth and very close to the surface of the oral mucosa, this stratified squamous epithelium, but it's not so stratified that the cells are keratinized at the surface. They're all still living cells with their nuclei in them. And so very close to the surface, you have nerves that are basically branches of the trigeminal nerve, the same nerve that serves the thermal pit. And it turns out these are also heat sensitive nerves as well. And this guy at Colorado University named David Chazar, he was at a meeting in Florida, a herpetology meeting, and there was a graduate student who was doing a talk on the histology. You guys know what the field of histology is about?
Tissues, sorry.
Yeah, it's the study of tissues and cells at a microscopic level, typically. So he was doing a histological examination of the oral mucosa of snakes. And he was running through his slides showing light micrographs of the sections of the skin on the inside of the snake mouth that he was looking at. And I think he was doing this in cottonmouths. And when he plunked up a cell that looked very much like a nerve cell, and you guys know what nerve cells are. Do you guys know what taste buds look like? You guys haven't had histology. We don't teach histology here any longer, right? None of you have had histology, correct? Yeah, okay. I didn't expect that you had. So let's go look up taste bud. When you look at taste bud structure, one of the things that you'll notice is they look like they're multicellular for one thing, and they look like little onions, little multi-layered onions. And the cells basically have these little sensory hairs that go out onto the surface of the epithelium. But then there are all of these cells that make up the body of the taste bud, and then there's a nerve that is basically taking the signals to the brain. And they don't look anything like a nerve cell. And this student had put up a photo of what very much looked like a sensory nerve cell. And if you know anything about sensory nerve cells, you'll see that these are what they call I forget what they're called, but they're one of two types typically. They're either a cell with kind of short dendrites and a long axon, or they're oftentimes these cells where they're pseudo-unipolar, where the nerve cell body is kind of away from the axons, both the dendritic and the dendritic axons, and what's the other type of axons that take it away? Afferent, I guess, axons. Basically, picking up the sensory information, taking it to the nerve cell body, and then sending it up to the brain. And so he presented something that looked very much like this, which is pretty typical of sensory neurons. And he called it a taste bud. But David Chazar, because he was a psychology professor and had taken courses in the medical school program, he knew that this is what a nerve cell looks like, and he knew that this is what a taste bud looks like. And so when he was presented with this image that the student was referring to as a taste bud that he was pretty sure was a nerve cell, he began to think, oh, this is how snakes do this. If these nerve cells that are in the nasal mucosa are branches of the same nerve that serves the pit organ, those are probably thermally sensitive as well, such that when the snake strikes, he no longer has contact with this specimen because his thermal pits are pointed towards the sky. But if he has mucosa that is covered in nerve cells that are also heat sensitive and go to the same spatial processing part of the brain as the thermal pit, you can actually track the moving warm-blooded animal with these nerves that are on the inside of the mouth. So he took this information back to Colorado, looked to find these nerves in his own snakes, and did these nerve tracing studies where you can inject a dye into a nerve and follow it back to where that nerve is connected in the brain, and basically found that these are all branches of the trigeminal nerve. They go to the same section of the brain and they have the same function. When I heard David Chazar talk about this when I was an undergraduate, he came to the department where I was an undergraduate and gave a talk. And I had already been taking courses in anatomy and things like that that are just kind of required in my curriculum. But then I wasn't really thinking about taking histology, but I began to see the importance of having courses that, even though I was training as an ecologist, might come in handy periodically. And so I began to see the value of other areas of biology beyond just ecological and natural history sorts of courses, because I was exposed to people like David Chazar, who was working on snake behavior, but used histological knowledge to give him insights into the behavior of pit vipers in this case. So I got to the kind of the end of my undergraduate career, and I had to go and get my degree plan approved by the dean of the College of Science at the place where I was a student. And I went, and you had to schedule an appointment with him, and you sat down and he kind of went through your transcript to make sure that you had everything that you needed to have. And he was flipping through my transcript, and he said, "Who told you could take 52 hours of biology?" Because we only needed like 36 or something like that to graduate. And I was like, well, nobody told me I couldn't do that. Or nobody told me that I could do that, but nobody ever told me that I couldn't do that. And he's like, yeah, you're really not supposed to take that much biology. He's like, what were you thinking? I'm like, I don't know. I just find biology fascinating and useful. And the further I've gotten along in my undergraduate career, the more that I find useful and applicable to things. And he just kind of shook his head and signed off on the graduation plan because I had exceeded the requirements for graduation, sent me on my way, and didn't realize that a year later I would be his graduate student. So once again, broad knowledge about a subject can give you insights into something that more narrow knowledge about a subject couldn't. And this is just one of the examples. And this thing that I was learning about when I was an undergraduate is now just commonly taught in textbooks, which is also kind of nice. That was a very long aside to get across the point that thermal sensitivity is not just limited to thermal pits. It's also found in other aspects of the snake, because that is actually what the combination of these things is what makes them capable of finding and tracking prey effectively. This is a thermal image of a pit viper in a box with a mammal. It looks like it's possibly a small rabbit from the looks of it. And one of the things that you see is that the snake is radiating heat. This is the colors are tied to temperature on this key over here. But one of the other things, what do you notice about the head of the snake?
It's the coldest.
It's cold. So one of the other things that they've discovered about having these thermal pits is that the circulatory system is also altered in the head of these snakes, such that it tends to shunt blood away from the area of the head where the thermal pits are located, so that the thermal signature of the snake itself isn't interfering with the thermal signal of things in its environment. So when we look at this adaptation, you might get tunnel vision in on the thermal pit itself without then recognizing that as the thermal pit has evolved, other aspects of the physiology of the animal has also evolved in a way that makes the thermal pit more effective than it would be if these other types of co-evolved adaptations had not also been in place. So one of the things that you find is that the snout of the snake is very cold because of reduced blood flow to that part of the snake, which then increases the sensitivity of the thermal pit to variations in thermal inputs around it, basically reducing thermal interference that might interfere with the function of the pit. So now there are people who study the circulatory system in the heads of snakes in conjunction with studies on the thermal pits themselves. And as you can tell, I totally geek out on stuff like this. So here we are in the reptiles. And this is going to be the beginning of kind of our taxonomic treatment of things. And so the first thing that we're going to be talking about is the Lepidosauria, which are the groups that include the Tuatara, which is the Rhynchocephalians. So we're in this part of the tree. The Rhynchocephalians and the Tuataras from New Zealand, and includes all the lizards and the snakes and the amphisbaenids. So before we jump into the Lepidosauria, maybe we should pause and say, as you've looked at the exam that's going to be over all that introductory material, the history of herpetology, the amphibians, do you have any questions about things that might be related to exam prep?
I have a question about how we're taking the exam, just because I'm not like at Jewel. Are we taking it
Oh,
on
yeah.
Zoom? Because I'm in a hotel, so I don't know about like how quiet of an environment I'm gonna be able to get in.
Yeah. So there during the time of COVID, there are all kinds of concerns. One of the concerns is an increase in cheating, which, quite frankly, I've never really had a ton of problems with cheating on exams because I've usually given my exams in class. Back in evolution and ecology, there was an instance of cheating that we caught with students taking exams remotely. But once again, it was like so poorly done. Like it just leapt off of the page and slapped you in the face as you were reading the answers. And so it was very easy to detect. So the way I'm going to administer tests in here is on the morning of the exam, I'm going to upload a PDF version of the test. You guys all have Apple Pencils and you have an iPad that you can use for doing things like I just did, which is, writing on a notepad. The other thing that you can do, and so you should prepare for this in advance, is I grade stuff nowadays by just having students, can you see that, or is it too bright? Oh, there we go. I have students submit their homework and things like this in PDFs so that then I can basically just go and grade them by making comments on the PDF. And I just scribbled all over Blaine Chapman's paper, but I will erase that before I give it back to him. And so what I will do is I'll put up a PDF, and then just whatever program that you normally use to annotate PDFs as you're reading primary literature and things like that, you should use that annotator. I use a thing called iAnnotate because I think it just does a really good job. I've been using it for well, since we got iPads, which was like back in 2015. You can write on your exam just like you would write on the exam if you were taking it in class. When you're finished with the exam, save that exam as a flattened file. When you save it as a flattened file, it prevents me from altering what you've done. So all I can do is continue to annotate over your annotations. Sometimes people don't save it as a flattened file, and I'll accidentally start writing something, and what I've inadvertently done is I've clicked on their thing and I end up erasing it, and then I have to click back and get their stuff to come back, and then I usually save it as a flattened file on my iPad so that I avoid messing up the work that they've done. I discovered that early on. It's like, oh no, I erased an exam answer, and so I had to go back and fix that. And then when I get it, I'll go through and grade it just as if it was a paper copy. And then I'll save my annotations that are the grading as a flattened file. And I'll write the grade at the top of the first page on that flattened file. And then you'll get that flattened file back as a PDF. And that's how you'll get your exam back. As far as the test taking environment, I usually open up a Zoom call so that I am online, so that if you have questions during the test, which people inevitably do, I'm available to you for that. But I don't require you to be online. I don't require you to have your video on, et cetera, et cetera. You guys know what the honor code is at William Jewell College. If I catch you cheating, you'll get a 0 in the class, and that'll be that. We'll just be done with that. So at the same time, I know you guys, and I can't imagine you guys cheating in my class. So I'm not also particularly worried about it. So I trust you. Honor that trust by behaving appropriately, and then everything's just good. So if you need a quiet environment, don't come on Zoom unless you actually have a question about the assignment. Sometimes when you open up the PDF, it might be garbled in the translation, in which case, I need to know about that immediately so I can get a different version of the test to you, depending on how your PDF reader that you're using is reading that PDF in. So that's the reason I'm on Zoom, is to answer questions and solve problems. I'm not on Zoom because I'm monitoring you for cheating, because I find that counterproductive. It sets up an adversarial sort of situation between professor and student that is counterproductive. And the other reason is I would prefer to think better of you than think worse of you, until you give me some reason to think worse of you, and then I'll start thinking worse of you. So yeah, I've talked to a number of people about this issue, and I would rather err on the side of thinking that people are good rather than people are rotten.
Okay, I just wanted to check because some of my professors have had us log on to Zoom, and since I'm in a hotel, it was going to be a little bit of a predicament.
Yeah, and if you're traveling for sports and stuff like that, that's just one more hurdle to get over in the process of taking the test. I want you to be thinking about the information that you know about herpetology rather than thinking about all this other crap. In my interactions with colleagues, we talk about all kinds of things, but we talk about students a lot. And I have some colleagues who their first response towards students is to think the worst of them. I have other colleagues whose first response is to think the best of them. I've actually find it more productive to think the best of people, expect the best of people, and then just proceed as if that's the way the world works until I have some evidence to start believing otherwise about that particular individual. It's just way more productive way of going about things. And it's certainly easier to engender trust and respect and things like that by adopting that. I've had students who just immediately think the worst of you. I'm like, think of what your life would be like if you switch that on its head and thought the best of people until you have some reason to think something bad about them. And certainly, you know, if repeatedly every time you see somebody, they're kind of constantly tearing you down, well, then you begin to form an opinion of that person based on their behavior. But if you walk into it thinking that that's the behavior that you expect, that's going to be the only behavior that you see, even if the behavior is not overtly anything like that. So trust. I trust that you know what the honor code is. I trust that you will abide by it, and I will maintain that trust until you demonstrate to me that I shouldn't have that trust in you. That's the long and the short of it for me. So no, if you have a question, bop on a Zoom, get your question answered, and you can bop off if you want. If you want, you can have Zoom running in the background so that you don't have to go through the hassle of connecting and everything, but just turn the sound off or whatever. I don't need cameras on. I'm just here to answer questions, same way I would be here to answer questions if you were taking the test in class. What else about the test?
Did you get my email
When did you send it?
last night or sometime yesterday?
Well, I didn't get it yesterday. Depending on when it was sent last night, I typically don't have my email running while I'm in class. Was it about the test?
Yeah.
Okay, let me go look for it. Oh, there is something. What is that? Oh yeah, about an interview. Yeah, I did see that. I didn't think of it actually in terms of the exam. Yeah, we can arrange a different time. Hang on at the end of class for a minute and we'll talk about it. Yep. Yeah, I saw the interview and then I didn't read further down because it also didn't register to me that, oh yeah, we have an exam that day. Yeah, not a problem. Other questions? All right, if you have content questions, my advice would be to study some for the test before lab tomorrow, so that when we're in lab tomorrow, you could ask questions about the exam as well as you continue going through amphibians in the lab. My advice to you in preparation for the lab exam next week would be to spend some time in lab working with one another, giving one another self-quizzes. So one person make up a quiz for another person, et cetera, et cetera. That's a good productive time for that last period leading up to the lab exam. I can talk to you more about kind of the structure of the lab exam and the procedure for the lab exam in lab tomorrow because there is a procedure for that as well. That has to be done in person, obviously, because you need the specimen in your hand in order to do that. So we only have a couple minutes left, so there's no point in starting lizards and snakes into Ataras in the last 4 minutes that we have here. So if there are no other questions about the test, you are free to go. And Rose, hang on just a minute. We'll work something out with the exam on Friday. Questions? Sarah's like,
Just a
I don't have
reminder,
any questions. I'm gone. Yeah. What's up, Kelsey?
just a reminder, I won't be in lab tomorrow. So
Okay.
I'll probably have you let me in the lab like next week once we're back, but I'll talk to you about that later.
Okay. They fixed the lock on the door, so now the door closes and things like that. So yeah, I'll need to unlock that for you.
Okay, great. Thank you.
All right. See ya.